Hello everybody. We are going to be walking through a StrainSEEK report today and Cannapedia, so folks can understand how to read these phylogenetic trees and what these reports actually mean. This is a medicinal genomics-based sequencing report. Now, there are two different flavors of sequencing at medicinal genomics, and soon to be a third. We had for the most of our history, we've been doing sequencing off of an 850,000 base pair target file, which was we generated that using restriction enzymes. It's a method known as reduced representation shotgun, and that has certain drawbacks and certain benefits. As we got more mature in sequencing, we are now just starting to roll out a system that does more like an exome sequencing in the plant, where we target 34 genes in the cannabinoid and terpene pathways, and then we tile 300 SNPs from the way we used to do it before, so we're backwards compatible to our old data, and then we also tiled in a lot of SNPs from the Phylos data set, from the Lynch data set out at Nolan Kane's lab, and from the Solar data set up in Canada. And by tiling SNPs and all those other systems out there, when we sequence a strain now, we can pinpoint its uniqueness amongst everything that's public. That's an incredibly important point that's going to be released in the coming system, because you can't do that right now. Everyone's sequencing different regions of the plant, and so you can't cross-compare the data easily because there's no overlap between the data. We want to change that and make it so you can sequence once and understand where you sit with all the other public data. So that Rosetta Stone part of the panel is really important, but I also think building these phylogenetic trees off of the genes that everyone's has under selection, which is the chemotype influencing genes, is probably more meaningful than doing genome-wide type of work. And then finally, we have had some people ask us about whole genome shotgun sequencing, and we do that on occasion. We just don't have the tools to thread that into Cannapedia just yet. So anyway, let's go to the phylogenetic tree to give you a sense of what this is. Here's a phylogenetic tree that's up on our site that is color-coded, and the color coding is important to see. It's down here. Red, orange, yellow, green, blue. This is kind of a Roy G. Biv thing, and the reds are clones. How we figured this out is that we sequenced lots of clones and figured what's the allowable amount of sequence variation we see between clones, and anything that's within a certain range we'll consider a clone. And then when you get into siblings and cousins, it gets a little bit more gray. The reason for that is that cannabis is such a diverse species that you can have plants that are tenfold more variant than humans are, and they can still cross. This is almost the equivalent of humans and chimps being able to cross and still have offspring. So if you did have something as distant as human and chimps that had offspring, you would expect to have very variable siblings. So a sibling in cannabis by no means it's very close if the parents are extraordinarily different. All right, so this is something that is a bit of a grayscale here. Even though we have it as color-coded, it is very difficult to discern what might be siblings that were made from back-crossed samples that were very homozygous, if you will. Those may produce very similar offspring, whereas if you take the analogy of the really most distant cannabis strains, like maybe a distant hemp line and something that's in a drug type, you might get very varied offspring. And so the term sibling can actually encompass quite an amount of genetic diversity depending on how different the two parents are. Nevertheless, this genetic distance map has all been switched to a color code to make it easy to view. And the important thing to note when you look around on this is the closest strain isn't always right next door in the phylogenetic tree, okay? And that's an artifact of making phylogenetic trees that is difficult to explain, but it's because there are so many different inputs to this that when you add in more samples and say each of these samples has 20,000 variants, simplifying that multidimensional data set into 2 dimensions can be challenging. In fact, even putting it into 3, 4, 5, some people like to put them into 13 dimensions just because there's that much texture to the data. But of course, when you start doing that, it betrays visualization of the human eye. It's very hard for the human eye to visualize things in more than 3 dimensions, and sometimes even putting it in 3 dimensions can often make it difficult to draw lines between 2 dots in that 3-dimensional system. Some of that is because when you start doing these principal component analyses, each of the dimensions doesn't always end up being the same scale, right? There may be log scales different between the first principal component, the second, and the third. And then when you start to chart that in 3 dimensions, well, you end up with a 3-dimensional PCA where each one of the axes is actually a different log scale, and your eye starts to imagine them to be XYZ coordinates, which they really aren't, and it can really create for some hallucinatory effects in the data. So what we've tried to do is simplify this in just 2 dimensions, but then also build tables that just put down the numbers. What is the genetic distance? Give me a hard number as opposed to a picture, so I can monitor my strain over time. And so we're going to walk you through how we do that. But do note, as you move around here, you'll occasionally find lines where the yellowest sample isn't always the one directly next door. It can sometimes be on the other side of the phylogenetic tree. Now, look at this one here, right? Lots of greens and blues that are in here, but you can see some of the yellows are scattered all the way around the tree. Why is that? Well, we're going to bring to these pages here. These are great sites we recommend people visit to understand how to read a phylogenetic tree and why these things sometimes radially split like this and why they get displayed that way. There's another one here at Nature that's worth reading as well. That talks a little bit about the history and what these trees represent. The lexicon about nodes and roots and the branch lengths here. So helpful resources, but lo and behold, as you add more samples to these trees, they will shift around in you a little bit. Usually you'll see the clones all still stick together. This blue tree clone, if we add another 100 samples to this cluster may actually end up over here, but it will still be clustered with all of its neighbors. It might invert that branch when it decides to draw the tree when you seed it with a lot more data. So keep that in mind. This tree will change. It's for really viewing pleasure, but if you want hard numbers, what you should really do is search for your strain and go to the report. So we're going to look up Congo. So Congo is a special strain for us because this is the last strain that Franco sent us. Franco, unfortunately, passed away chasing down this strain and many others, actually. This was a horrible event. He was a huge contributor to the field, and what you're going to see is he really was a unique strain hunter in that he was able to find some of the most unique material. And this is probably the most unique thing we actually have in our database. Okay, so this in September they sent to us. They were not able to get the terpene and cannabinoids done. They probably have that now, but they at the time did not submit that with the DNA sample they shipped in. So we don't have that information on the strain, but we do have a lot of other interesting information on the strain. You can go down this report, and if you want, you can click on this. This will bring you back to the phylogenetic tree. Now, if you want to find your strain on here, hit Command F if you're on a Mac, type in Congo, and you'll get Congolese. You'll also get, where else? Congo down here. And you'll see this one here has almost nothing else that lights up on the tree, other than maybe this hash sample over here, which is blue, by the way. But you really don't see anything else lighting up when you get down to what this is. And you're going to see the genetic distance table shows this thing to be far away from pretty much everything. Now, let's look at the heterozygosity here. This is an important number. This is basically a representation of how distant your mother and father genomes are in the plant. This is how many bases have single nucleotide variants on them. And usually in cannabis, this number is more like 1%. So this is at 0.8 or 0.79. That means it's fairly inbred and more homozygous than most strains that we see. This number sometimes gets as high as 1.3, 1.4, and that just really tells you that that's the number of bases that differ in the plant. 1 in 100 would be a 1% heterozygosity number. For perspective, humans are 1 in 1,000. Okay, so that's where that tenfold difference in variation comes from. And then this file here is incredibly important. Okay, this is the VCF file. This is a variant call format. It's a collection of all the coordinates and all the contigs in the genome where we have an actual variant, and what we call a SNP. And this file, if you click on it, will download you probably a file that's somewhere probably around 50 meg or something. Now, this file, let's see if it will click and download. What's it say? Yeah, 47 meg. There you go. This is the most important file for intellectual property reasons. This file and these two FASTQ files. This file is the one that we utilize as a fingerprint for the strain, and we actually perform a hash digest on this sample and put that hash digest into the Bitcoin blockchain system. We do that because that's an immutable ledger that can never be reversed, and that puts a cryptographic hash of your data in the blockchain so that you can prove that your file existed at that time. It doesn't put your 47 meg file in the blockchain, so we highly recommend you download these files for your own record keeping in case our site for any reason goes away. This file is really the key to unlocking that hash that's in the blockchain. Now, the hash in the blockchain cannot create your file. It's a 64-byte string that no other file in the world can create that string but your file. So the fact that that's in there at that timestamp means, and you can present, when you run SHA-256 in this file, it produces that fingerprint or that hash that's in the blockchain. You can prove that your file had to exist at that time because no other file in the world can produce that actual fingerprint. That's the unique nature of SHA-256. They're collision-free cryptographic hashes. Okay, so if you change one letter in this file, it will give you a different hash, a completely different hash. And so that's an important thing to remember. Now, these FASTQ files are the actual sequencing data. If you want to download these and do your own analyses, we encourage it. That's the forward read and that's the reverse read. And this is what's utilized to get aligned to the genome and compared to everything else we have in the database. Here's the Congo sample, and you can see this clone distance here. We have it's a little bit of a grayscale here because we're still getting more finer and finer resolution as to when siblings turn into clones and vice versa. But this thing is as clonal as can be with itself, obviously. And then you look at, is there anything else like it in the database? And there's nothing that looks like a clone, nothing that even looks like a sibling, and everything is way out here at 8, 7.87 or 8. This is really one of the largest genetic distance spreads we typically see. We're usually seeing numbers in the 4 and the 5 range with samples that are in North America. And this thing caps all the way out at 7.87 with Canopy. Now we have two different data sets that we compare these to. There is the nearest genetic relative to everything we have in our database, which is the first thing we represent. And this is a dynamic list because we're getting samples in every day. And so this list gets longer and longer and longer. And that annoys people sometimes because they bring a sample to us 6 months later and suddenly we're not comparing it to the apples that they compared it to before. And so everyone asked, hey, can you get us a set of like standards? What we call a base tree, like where you always are analyzing and comparing to this exact same set of samples. That way, when we come to you year after year, we can see whether there's been any drift. And this is what we call our base tree. And there's much fewer samples here. But nevertheless, you can also see there's nothing in this base tree that comes anywhere close. Cherry limeade is the closest thing. Okay, so you can click on any one of these and hop to those reports if you want to explore outward and get lost in all the interesting strains that are out there. This is a very interesting one we always find. This one is really dark, probably a lot of, if I remember correctly, this one I thought had a lot of anthocyanin in it, but maybe not. Maybe I've got that one mixed up. Anyway, we're getting diverted. Okay, let's finish this off and look at what the actual blockchain means. Okay, so this QR code, if you click on this actually is a QR code for this site. So if you want to put that on a label, on a package anywhere, someone can scan that with their phone and it'll bring them right to this page. So that is a QR code that self-references this URL. This is the SHA hash of your VCF file, okay? And that is sitting inside this blockchain transaction, which you can see was done 10 months ago. And at that time, it cost us this many Bitcoin to do it. That turns out to be a lot of money these days. But nevertheless, this is basically proof of existence that this data exists in the blockchain at this time stamp, and only this sequence of information was actually embedded in there, and that's actually a representation of the file right back here. That SHA hash is what your VCF file will produce if you ever run SHA-SUM on a command line in a Macintosh or any other computer. Should usually have SHA-SUM built into the OS, and it will just spit out this as an output if you give it the VCF file as the input. Okay, so that's kind of the story on these StrainSEEK reports. Please leave us feedback and send us an email if you have any other questions. We've got a lot of partner labs that help provide this terpene information. We do incentivize people to provide the terpene information. You'll see this in a lot of the other continuing customers that are here, because there's actually a credit system. You'll get, I think it's $50 off if you provide the terpene information. It's a credit to another StrainSEEK, not hard cash, but anyway, we want you to make sure you get those measured and put it in here, because the information of these terp measurements is obviously one day going to associate with some of the genetics, and those will be very interesting markers for everybody and will help everyone with their breeding program. So please check out Cannapedia. Enjoy the Philo tree. You'll see some new versions of this coming out as we roll out the newer panel. The newer panel that does this microexome on the cannabinoid genes, really important, and it is producing beautiful data. It is actually sequencing 3.2 million bases in the plant. So by our estimates, that is 25 times more than you can get done anywhere else in the cannabis space right now. So it's really the largest, most comprehensive sequencing panel out there, and it's It might be a little bit more pricey, but it is well worth it if you're trying to split clones and siblings. If you're doing anything in the intellectual property arena, you do not want to cut corners and sequence less. It becomes increasingly harder to split clones and siblings if you sequence less than when you're sequencing more. So it's just obvious logic there. You sequence one base, you can't discern the difference between many plants at all. You move that to 10, 20 bases, you get much more signature. You go out to 3.2 million bases, and suddenly you're dealing with enough sequencing density to do whole genome association studies, which is where marker-assisted selection is going to go. And this is how we're going to pull out drought resistance and powdery mildew resistance and all those fascinating features of the plant, is by sequencing enough density where we have, on average, a marker about every gene, and that puts you up above a megabase in sequence. So enjoy, everybody. Thank you.